Welcome back, friends. Now, in previous discussion, we just started about the hypersensitivity, and we've discussed the basics and introduction of hypersensitivity and what is hypersensitivity. And we have also learned that there are four major type of hypersensitivity that came to be existed. Now, among them, in this particular video, we'll be talking about the type 1 hypersensitivity, which is much more we need to learn in much more detail because it is the most common one and it is also dangerous one. So we are talking about here type 1 hypersensitivity in this particular video. Okay, let us change the color. It will help us to understand it. Type 1. Okay, type 1 hypersensitivity. Now, in this case of type 1 hypersensitivity, there are cells which are involved. This is a cellular mode of hypersensitivity, and also chemical mediators will come in this particular hypersensitivity. Now, and also antibodies involved in this kind of hypersensitivity. Now, let us look it as a whole story to learn what is type 1 hypersensitivity. The major key player of this hypersensitivity is mast cells. Now, what are these mast cells? Mast cells are large cells. The cells are having granules, and the granules are contain all these small chemical mediators like histamine, heparin, serotonin. Okay, and they are having many different kinds of granules. They are actually chemical factories, so they can produce chemicals and they pack them into small vesicles and just throw them outside the cell, and those chemicals is going to have different effects. Okay, for example, chemicals like histamine can have effect like vasodilatation and airway constriction, smooth muscle contraction, and so on. They are also having potent chemical mediators like leukotriene B4, leukotriene C4 and D4, something like that. So these are or arachidonic acid pathway mediators. Okay, so these chemical components will be released from these mast cells, and those chemical component release will have different effect into our body. Okay, so let us first understand that. So in this case, we must know. So let me draw a structure of a mast cell. So say this is our mast cell. This is our mast cell, and this mast cell, say let us draw a nucleus. This is the nucleus, huge nucleus inside the mast cells, and inside it we are having small vesicles. We are having two or three different types of vesicles actually. So let us draw two different types of vesicles here. One is red one, another one is the black one, and all of these vesicles are having small dots. So these dots are actually chemicals that are present in these small vesicles. So this is our mast cell. What is it? It is, sorry, let us write mast cells. So yeah, now mast cells are chemical factories. Now they have these chemicals as granules inside their body, inside their cells actually. Now what will happen in this case, the response or the hypersensitivity response that we are going to see from this type 1 hypersensitivity is that release of these granules outside. So it will end up with release of these granules outside. Okay, so the release of granules, the granules like histamine and also heparin, serotonin, and other granules like leukotriene B4, leukotriene C4, and leukotriene D4. Okay, so these things are lipid mediators, and the previous one are the protein mediators. Now these are the mediators that will come out from these mast cells when the mast cells will be activated. Remember, when the mast cells are activated, these chemical mediators will come out, will spill out outside. Out means where the mast cells reside. It resides in our tissue. It resides in tissues. Now mast cells are in tissues. It will release these mediators, histamine, heparin, leukotriene B4, C4, and D4. Now these mediators, serotonins, heparin, they will go and start damaging our tissue. what kind of damaging effect that they can have? The answer is this histamine has two major effects into our body. The effect is vasodilatation, vasodilatation, and second thing they are having is smooth muscle contraction. So vasodilatation and smooth muscle contraction. These two things, most of the cases, this histamine is the major component of this chemical weaponry. Now this histamine can lead to two different, vasodilatation and smooth muscle contraction. Vasodilatation means, suppose we are all having this venules and all these regions. So as vasodilatation is happening, so the permeability between the cells of venules, the endothelial layer, is getting increased. Now as the permeability is getting increased, so cells start contracting. Starts contracting, permeability is getting increased. So the blood will start to come out into the tissue. So as a result, it will start to swell up. This is one thing it is happening. And second thing is a smooth muscle contraction. Smooth muscle, wherever the smooth muscles are present in our body, it start contracting. For example, smooth muscle of our bronchiole. Now as they start contracting, the smooth muscle contraction as a result lead to the airway constriction, right? So as smooth muscles start to contract, it will create immense force onto our trachea and it will lead to airway constriction. So we cannot breathe at those times. So stop breathing and also vasodilatation can often lead to death. Okay, so that's why they are very important. And also leukotriene B4, C4 and D4. This leukotriene B4 can call upon other cells of immune system. Look what I've got, just come and join us. Okay, so the effect of leukotriene B4 is it is acting like a uh, chemotactic factor chemotactic factor for neutrophils, basophils, and also chemotactic factor for other cells like macrophages. So it call upon all those cells in this case. Okay, and leukotriene C4 and D4, these things are also potent mediators for smooth muscle contraction and vasodilatation. Okay, so as a whole, if these granules are fused, these vesicles are fused with the cell membrane of a mast cells, it will create hell of a mess, right? It is creating hell of a mess inside your body. Now it is taking all the immune responses on its own. So it's a bad cop. So whenever it finds something, it is creating a situation where it call upon all the cells and secreting these chemical mediators that are going to kill your cells, kill your own cells. So the tissue cells start to hamper, start to get die. Okay, so in this situation, we definitely don't want this kind of situation. So this is a hypersensitive reaction. This reaction is not wanted. In general process, this reaction must not be happen. Okay, so we need to prevent that. So in type 1 hypersensitivity, these things are happening. Now, how these mast cells get activated? Remember, I've told you something, that these things will happen only if the mast cells are activated. So this is, if this is inactive, all those things will be shut down. But when the mast cell is getting activated, then these things are going to happen. Now in type 1 hypersensitivity reaction, these mast cells are getting activated. Okay, now how these mast cells are getting activated? Now let us go back from this story, how it is getting activated. For the activation of these mast cells, it needs some signaling inside the nucleus, which is telling the cell now it's time for fusion of the granules with the membrane. Okay, so which is giving the signal? What is giving the signal? The signal is provided by a mechanism of signal transduction. Okay, by mediation, by the help of an antibody, IgE. The antibody called IgE. Remember this antibody, IgE, or Immunoglobulin E antibody, which is helping to trigger the response inside the mast cells, that it is the time for fusion of these vesicles with the cell membrane. It is the time for getting activated. Okay, now how these mast cells are getting activated? Now the mast cells, we have seen the inside story of the mast cells. Now let us look the outside story of the mast cell. The outside story of the mast cells are telling us that these mast cells are having receptors. Now they are having receptors for, say, something like that. Receptors. They are having receptors for immunoglobulins. They are called immunoglobulin receptors, IgE receptors. So these are called IgE receptors. It is IgE receptor. Okay. Or this receptor is also called Fc epsilon receptor. Because the IgE in any kind of immunoglobulin, it is two different regions. One is the upper region, which binds with antigen or antigen binding region. Another one is the structural integrity region, which is the below or bottom part. Okay, so if we draw the structure of a antigen, it will look something like that. This upper region is for binding with antigens, but this is the part which is called Fc. This is the part which is called Fc. Now the receptor that are present, not Fc. Fc is the part here in the this IgE, but this receptor is called Fc epsilon receptor. Okay, so Fc epsilon is the receptor for the Fc region of this IgE antibody. Now IgE antibody can go and sit onto this receptor, and then it can trigger some secondary messenger system to go and tell the nucleus, tell the gene that it's a time for division. It's not division, it's a time for fusing these prepared granules with the cell membrane and start secreting these chemical machineries. Okay, now how it is happening? Now the general mechanism for that is, the general mechanism is that two IgEs are required. So say here comes it, 1 and this is a 2. 2 IgE are required. 2 IgE is coming and sitting onto the Fc epsilon receptor onto the surface of the mast cell, and it is also requiring, still at this point it is not activated, though IgEs are present, they are going and sitting in on it. So 2 IgE are required. Remember, it is sitting onto the Fc epsilon receptor, Fc epsilon receptor here. So let me write it here. It is Fc epsilon. This is, and these are IgE. So 2 IgE are coming and they are sitting onto Fc epsilon receptor, but this is not it. This is not yet done. It's need to be cross-linked. This IgE are need to be cross-linked with each other. And what makes them cross-linked? Here comes the interesting part. It is the allergen which is the responsible for cross-linking these IgEs with each other. Now what is an allergen? Allergen is a substance which can create hypersensitive reactions inside our body. Though it's not pathogenic for us, though it's not harmful for us, but still it can trigger our immune response in some manner or what. Now another question can arise, that whether this allergen can create hypersensitivity in all individuals. No, it can create this hypersensitivity conditions only in few fraction of a population, not the whole population, only a particular fraction of a population. Okay, so if actually it's suggested that 20% of the population can have this kind of reactions. So 20% of the population can have a hypersensitive reaction against a particular allergen, and 80%, remaining 80% of the population will not have any kind of reactions. Okay, now in this case again, these IgEs are sitting onto the IgE receptors, Fc epsilon receptors. Now allergen will come. Now suppose this is a person who is susceptible to allergy. Now allergen is coming in. So allergen, for example, say this is a structure of an allergen. Oh, just looking like a sunflower. It is coming and attached. Now this allergen, one allergen, it is cross-linking 2 IgE sitting onto the Fc epsilon receptor onto the surface of the mast cells. And as it is sitting here, it triggers, it sends a signal and that signal in turn rises. This signal in turn rises the cyclic GMP count inside the cell. It rises the cyclic GMP concentration inside the cell. So we are having guanosine monophosphate. Now it is cyclized to produce cyclic GMP. Now the concentration of cyclic GMP inside the cell is getting high. Now as a result, it can give several, this is acting like a secondary messenger. It will send the signal to the nucleus. Now it's time for fusion of the granules. Now it will rearrange its cytoskeleton. Rearrange the cytoskeleton of a cell, because each cell are having a cytoskeleton on its own. Remember, they are made up with different proteins called tubulins in case of eukaryotic system. Now those tubulins start to rearrange between themselves. It is having the signal now. Then these granules will start migrating. Migration starts. So the granules start migrating, and then it start to fuse with this. As it is fusing with it, it start to release these chemical mediators like histamine. So these things all are triggered, or all are on. Now as a result of on nature of all these things, start secreting chemical mediators, and we can see the vasodilatation, smooth muscle contraction, and these effects. Okay, so for the effective set for hypersensitivity, type 1 hypersensitivity reaction, we need the huge amount of production of IgE, because we are seeing only one receptor. There are 1,000 receptors placed in the surface of the mast cells. All need to be activated to get a strong signal which can trigger the cell. Tell the cell now it's the time for go on to the hypersensitivity reaction. Now we need a lot of IgE production, and also we need a lot of allergen. Okay, now allergen could be a pollen, it could be animal fur, it could be dander or something like that. Now let us go back. Now what is the production unit for this IgE inside our body? Now there are B cells. Remember, B cells. B cells, when they mature, they produce plasma cells. Now these plasma cells, so remember, they are huge cells, big nucleus, and they are producing all these IgEs. So let us draw IgEs like that. So these are called the plasma cells. Plasma cells. Now B cells converted to plasma cells. Okay, now these plasma cells are antibody secreting factories. Now these particular plasma cells are secreting lot of IgEs, because it is destined to produce a lot of IgEs in this case. Okay, now for catching type 1 hypersensitivity, the individual must have lot of mast cells who are destined to produce IgE. Remember, they must have lot of plasma cells who are destined to produce IgE. That's why all of these IgEs can come and sit onto the mast cells and can start doing this type 1 hypersensitivity. Now why some of the people is having this plasma cell producing a lot of IgE, some of the cells are not, some of the people are not? This is due to a mutation. This is due to a mutation. The mutation that happens inside our chromosome, as a result of that, some of us are having cells, T helper cells, T helper 2 cells. Those T helper 2 cells, as they are getting much more T helper 2 cells in their body, so as a result, these individuals will have, for example, for a mutation, this mutation will trigger to produce a lot T helper 2 cells. Now as they are producing a lot of T helper 2 cells, this T helper 2 cell is going to trigger B cell. They are going to trigger the B cell to be activated. Okay, so they are having high concentration of T helper 2 cells, so they are getting a lot of B cells. Now these B cells will destined to produce a lot of IgE. Why? Because these T helper 2 cells are telling all the other cells, all the other totipotent or pluripotent cells, now start producing B cells, don't start producing other T cells, start producing B cells. Now why? Because they are secreting some chemical mediators, and those chemical mediators are interleukin 4 and interleukin 5. Now this interleukin 4 and 5 combiningly together tell all the B cells to convert and to produce IgE instead of any others. Generally, B cells, when they start to produce, when they start to grow in bone marrow, then they are trained, they are required, they start producing IgM. Okay, they start producing IgM, or sometimes they produce IgE antibody. This is quite natural to produce IgM or IgE. But if they get the signal via IL-4 and IL-5 from Th2, T helper 2 cells, it is telling them to develop IgE instead of IgM, to develop IgE instead of IgG. So they are start producing IgE. So as they are producing IgE, this IgE are in higher amount in the person's blood, it will go and bind to it, then allergen will bind, then all the system will work. So now let us go back from this situation. So suppose one person, he is having this mutation into their chromosome. Now as a result of that, its T helper 2 is secreting IL-4 and 5 and telling the B cell to go and produce IgE. Now their mast cells are producing IgE. Now lot of IgE inside their blood. Lot of mast cells are also inside their blood. Now what will happen when sometimes a allergen comes in? They are sensitive to the allergen because they are having a lot of Ig and lot of mast cells. So now those Ig will come and sit onto the Fc epsilon receptor. Allergen is coming and it is also cross-link these two antibodies. Now it will give the signal to give rise this cyclic GMP concentration. As it is rising cyclic GMP concentration, it will rearrange the microtubule system, rearrange the cytoskeleton system inside the cell, and as a result it will start fusing these granules to the cell membrane. Then it will start releasing histamine, heparin, leukotriene B4, C4 and D4, and then histamine start to vasodilatation and smooth muscle contraction. And on the other hand, leukotriene molecules will start getting chemotactic factors and they are start calling neutrophils and basophils and also eosinophils. Okay, so as a result they are creating a lot of system, they are creating the immune response, they are creating the area where everything is present starts secreting, starts swelling out, start getting red, and then all the rashes come out and something, all these things are happening. This may be small, the effect may be sometimes slower, sometimes also in some individual the effect could be life threatening. Sometimes, especially if you are having asthma or something like that, it could be life threatening because smooth muscle contraction can lead up to the airway constriction. Okay, so this is the overall process of type 1 hypersensitivity reaction that we can find in the most of the cases of our individuals, and I hope this will help you. Thank you.